Assalamu alaikum. Can everyone hear me? Okay. Assalamu alaikum. I'm delighted to be here. And today I'll begin this session. I'll be talking about my graduate work where I've been exploring one of the biggest public health problems, which is known as antibiotic resistance, and how I've used a molecular and evolutionary approach to trying to combat this issue. In my talk, which is called Combating Antibiotic Resistance, Evolution Strikes Back. And to give you in context, infectious diseases have ravaged human health for ages. In fact, we see just over 100 years ago, 50% of the mortality seen in the world was due to infectious diseases. Even in the 20th century, we saw the rise of smallpox, which was a huge pandemic across the world. We saw outbreaks of cholera and other pathogens affecting so many poor communities in the world. And even here in London, we saw cases such as the situation here where hundreds and thousands of school-age children were affected by tuberculosis. And we see that today, infectious diseases, for most part, we've been able to combat this issue, and it's not as major threat. And this is primarily due to many other treatments, but primarily due to rise of antibiotics. And antibiotics are, in fact, compounds or natural compounds produced by microorganisms to fight off other microorganisms. They're, in essence, weapons used by bacteria to fight off other bacteria. In humans, we've been able to use this power to treat many infectious diseases. And for instance, in World War II, we saw the rise of penicillin, which played a huge factor in treating wounded soldiers. But today, in just recent times, I'd say within the last decade, we've been infected by a true crisis. And you may have heard of it in the media as well. We're finding that more and more bacteria are becoming resistant to these drugs, meaning that when a person goes to the hospital, there's no effective antibiotic that can treat that infection. And we're seeing the rise of superbugs, which are completely resistant to all known antibiotics. And just this past year, we saw the rise of something known as the superbug, and sometimes called the phantom menace, where every single antibiotic is ineffective against those drugs. And the UN has declared a major emergency, describing how if this continues, then we'll be in a post-antibiotic era, where we'll see setbacks in modern medicine. So the question I wanted to ask in my project, in my graduate work, was to understand how are bacteria able to do this? How are they able to share this resistance? And what's allowing this resistance to spread? And to ask that question, we have to understand something basic about the biology of bacteria. And it's a bigger, broader question of what really allows bacteria to become pathogenic. For instance, on the left, we see that this bacteria here is completely harmless. In fact, it's even beneficial for us. In the human body alone, we see that there's 10 times more bacterial cells than there are, in fact, human cells. And bacteria play a critical role in our ecology. So what allows this bacteria to make a very slight jump, rapid jump, to become a bacteria like this, which can cause pneumonia and so many other pathogenic illnesses? And to understand that question, what allows this rapid jump to occur, we discover that bacteria possess a novel capability of evolving. It's very unique to bacteria, and it's known as horizontal gene transfer. And this allows bacteria to acquire genes from the environment, from other bacteria, or even viruses, using those genes and incorporating in for themselves. And this is how drug resistance is also spreading, where bacteria are able to acquire these foreign genetic elements. But in fact, we know that this is actually quite a dangerous process, because if you can imagine just taking any random piece of genes or DNA from the environment and putting it inside themselves is a very dangerous process. Those genes can really disrupt how the cell works on its own. And we find that most often than not, horizontal gene transfer is a very dangerous process. It's quite costly. So bacteria have to find a way to actually mitigate this process, determine the benefits of acquiring these genes, but also prevent any drastic consequences that can occur. And I'll be showing today how some bacteria, a very select few bacteria, have a way to actually resolve this issue. And it's been done through this protein known as HNS. So this protein HNS, and I'll be showing here, it's found in a very select few bacteria, and this is, I'll be giving an example of salmonella. Salmonella is a pathogen which causes typhoid fever. So in fact, if you take the chromosome, and this is just the chromosome of salmonella, and if you find where those foreign genes are found, so salmonella on its own has many accumulation of foreign genetic elements. Anytime you see a peak, it represents an accumulation of those foreign genetic elements. And if you wanted to look at the binding of this protein, where is this protein actually binding to the chromosome? And we find that there's a very strong association. Where there's these aggregation of foreign genetic elements, that's where this protein HNS binds. And this is highly interesting. And what allows this protein to do is actually control this process of horizontal gene transfer. And it does this by binding to it and then ultimately silencing it. So just to give you a model how this happens is that, so bacteria such as Salmonella acquires this foreign gene, let's say from a virus. Regardless if that gene is beneficial or not, then that gene is uptaken by the bacteria and put inside its chromosome. Immediately it's recognized by this protein HNS, and it can silence its expression, make sure that's not turned on at an inappropriate time. But in fact, and this actually allows it to mitigate those costs. So I mean, it can acquire those genes without any negative consequences. But in fact, we know that this protein HNS is only found in a select few bacteria. We don't know how the vast majority of all these pathogens are acquiring drug resistance, how they're able to do this process. So the project that I wanted to explore was quite simply, how can bacteria without this protein HNS regulate or control horizontal gene transfer? And to do this, I use a molecular approach of quite simply where we take a model where we know HNS plays a critical role, we delete that gene, and then observe how the cell can respond in an experimental evolution approach. So simply, first thing I did is delete this gene. So this is just a growth curve. On the x-axis, you see the time, and the y-axis is a measure of density, how well the bacteria is growing. So if you take wild-type bacteria or something like a regular bacteria without all its genes intact, you see that the bacteria grows quite well. As soon as you delete the HNS, as you can expect, because it controls such a large amount of genes, as soon as you delete it, this bacteria is quite sick. So what I wanted to do next was, okay, let's try to use experimental evolution where we propagate these bacteria and observe if they can compensate for this, if they can evolve some way to re-control these foreign genes. And if we can find any mutations that do this, we may get insight into how bacteria without HNS actually control this process. What's the ancestral mechanism? So to do this, I use automated robotics to do this. In essence, when a bacteria grow to a certain level, then they're repropagated to the next set of conditions. And in essence, what you're doing here is we're selecting for the fastest growers from here to re-continue on. And what you expect them to do is that they'd be able to grow faster and faster. They'd be able to, this time between each interval will get quicker and quicker. Just to give an example, this is the mutant bacteria. They use the ones that don't have HNS. And this is wild-type bacteria as a comparison. They're quite sick, meaning that there's a very strong selective pressure against these bacteria to acquire something to compensate for this. So when we do this, now if I take this time from one jump to the next jump, and if you plot it on the y-axis, on the x-axis is generation, or the time of how long I evolve these bacteria, we see that there's a drastic reduction in this time to reach between one passage and the next passage. In essence, the bacteria are getting faster. And we did this for over 500 generations. Now, if you wanted to do this in human years, this would be thousands and thousands of years. So this is a powerful tool that we can use. So now we know that bacteria are getting faster. They don't have this protein HNS. Question is, what's allowed them to make this huge jump? What's going on? What, at the genetic level, what allowed them to do this switch? And to do this, we use whole genome sequencing. We're able to read the genetic code of these evolved pathogens. And in fact, we don't just do this experiment once. We do it repeated times to see how evolution repeatedly acquires these mutations. What are the common mutations that repeatedly occur? And when we do this sequencing, we in fact find that we're going to get a long list of mutations, hundreds of mutations that naturally occur. But most of these mutations, they only occurred in one experiment. So they're not as relevant to us because there's just random mutations. They may not have any impact as well. However, it becomes really interesting when we find repeated mutations, meaning that all of those different experiments, if they all have the same mutations, those become extremely important hits. So we find that one gene, this gene known as Rho, repeatedly acquires mutations in all these 5 experiments. So what's this gene Rho? What is it actually doing? Now, it turns out it's a highly important gene. And unlike this protein HNS, which was originally found just in a few select bacteria, HNS is found in actually 90% of bacteria found in the world. So that means those bacteria in your gut, those bacteria out in the environment, and those bacteria, even those found in extreme environments of the world, they all have this protein HNS, showing that it's critical to the very basic biology of bacteria. And it's generally thought as being as a housekeeping gene, meaning that it's involved in just the regular upkeeping of bacteria. And what's primarily known as doing is, what was originally thought of doing, is just reading the genetic code and allowing for bacteria to control when genes should be turned on and turned off. Because you can imagine bacteria don't want to waste resources. They only want to express genes. They only want to turn on genes at appropriate times. So the mutations that I found that repeatedly occur, we actually found they're all closely localized to the same place. And they're all affecting kind of the same domain of this gene. And through a series of other molecular experiments, I tried to elucidate and figure out what are these mutations actually doing. And today I'll just present a model of what we believe how this protein is actually playing a role in horizontal gene transfer and bringing it back to the rise of superbugs. So if you can imagine what happens in superbugs, you have this one resistant bacteria, and all the other bacteria are able to be killed by antibiotics. However, this resistant bacteria, it has this genetic element that it can transfer to other patients or other bacteria. And to mitigate this process, we know that in select few bacteria such as Salmonella, we have the protein H and S. But in fact, H and S is working under the umbrella of this protein Rho. And we believe that the majority of bacteria actually work through this Rho protein to facilitate horizontal gene transfer. And what we know is that these foreign genetic elements are actually exploiting this Rho protein, exploiting this function that bacteria have to regulate genes to incorporate these foreign genetic elements. So now if we can design drugs against Rho, we're not going to be able to kill bacteria, but we'll be able to limit their spread. We'll be able to limit how quickly they can spread their resistance and other virulence genes to other bacteria. So in conclusion, I showed here today that horizontal gene transfer is an important role in bacterial evolution. And using experimental evolution and using principles of wisdom that evolution must be occurring through some wise process, I showed that this protein most likely this protein Rho is playing a key regulator in horizontal gene transfer. And if we design drugs against Rho, we'll be able to stop this process and also solve this crisis of drug resistance. Jazakumullah, and I'd be happy to take any questions.